Hi, I'm Lechuan Li from your lab of Rice University, and today I'll introduce our work joint embedding of biological networks for cross-species functional alignment. Our method introduced a new way to realize the functional relationship between genes across different species, especially between human and different model organisms. As we know, model organisms are very important for study human diseases, but there is a gap between human and model organism biology. For example, if we want to study a specific gene for a certain disease in human using mouse as a model, how can we find the genes that carry out the similar functional roles in mouse? And the problem also exists other way around. If we already find something interesting in mouse, how can we bring those knowledge back to human? So it's very important to achieve this functional information transfer between human and model organisms. Actually, their researchers has already used the orthologous genes as a link between different species to transfer knowledges, to transfer informations. And the orthologous genes are the genes in different species that has only diverged as a result of speciation. But there are two main shortcomings directly using the orthologous genes. The first is only limited number of genes have orthologs, and depends on the evolutionary distance, the number also varied between different pair of species. And also, currently, people are determining the orthologous information through sequence similarity. And the sequence similarity cannot guarantee the functional similarity. So the orthologous pairs are imperfect. So people then decide to incorporate more information to get a better inferring for functional information. One of the information that are used are protein-protein interactions. Proteins work together in organisms to carry out functional roles. So the proteins that are physically interact with each other tend to have same or similar functions. Therefore, people construct those protein-protein interaction networks, which individual nodes are proteins, and edges are whether two proteins have a physical interaction or not. Having protein-protein interaction network for each individual species and analyze the network topologies can help people inferring the functional relationship between genes across different species. And also, we still want to use the orthologous information I talked about before. So these two kinds of information are the main information the previous existing global network alignment method uses. And there are also lots of existing methods on that direction, and they use different types of strategy to determine the network topologies. But they all have one common shortcoming, is that they combining the network topology information and sequence information linearly. And we can see from the right example here, sometimes those two kinds of information pit against each other, which indicates there is a more complicated relationship between them rather than a linear relationship. So more recently, people trying to use a general purpose embedding approach to better capture the relationship between network topologies and orthologous information. The idea is that when we have two protein-protein interaction network, and the stars in the figures indicates the orthologous genes between two species, we will first have individual embedding for each of the network. Then using the orthologous genes as anchor links, we can create a joint network embedding that have genes from different species in a same latent embedding space. And one existing approach use this kind of idea is the Monk method, but it has two major shortcomings. The first is their embedding dimension is the size of the PPI network, which for biological networks is usually in a scale of thousands or 10,000. This number is too large for downstream analysis. And the second is their alignment is directional, which means if we're aligning human to mouse and mouse or aligning a mouse to human, it will give different result. And this is also annoying for downstream analysis. So we want to design a method that can handle the sparsity of biological network and also capture the nonlinear relationship between topological structure and sequence formation. Then it should have a bidirectional mapping in the alignment, which means it will give a consistent result for a certain species pair. And finally, we want the embeddings with customizable dimension, which is good for the downstream analysis. Therefore, we propose our method embedding to network alignment. We call it EDNA, which have mainly have 3 steps showed here. The first step, we use an autoencoder to create individual network embeddings. Then when we have 2 species with 2 autoencoders, we use an idea inspired from the natural language processing field that swaps the encoder and decoder for 2 species to align the individual network embedding together to a joint embedding space. Once we have the joint embedding, we can use a cosine similarity to determine how similar genes from 2 different species. So I will explain our method in more detail. The first step is individual species network embedding. And instead of directly using the adjacency matrix as input, we use a more complicated kernel, the NetMF matrix, which is existing approximation, deep work process approximation. The idea is that we can simply consider each entry of the NetMF matrix as the number of passes with smaller or equal to a windows like a hyperparameter t, a window size t, for 2 given node in the network. So in this way, each entry of the matrix is not only whether they are connected directly by an edge, but it's a number of passes between them. So it's considered more further neighborhoods in the network instead of only the direct neighbors and 2 hop neighbors that in the adjacency matrix. So in using the NetMF matrix as input, we'll feed it into an autoencoder. The autoencoder contains 2 parts of loss functions. The first is the global structure loss, which is achieved through the reconstruction using the autoencoder. And the second part is we want to emphasize the similarity of the embedding between nodes that are directly connected by an edge. So we are forcing the nodes that are directly connected by an edge to have similar embeddings in our embedding space. And together, for those 2 parts of loss, together with 2 regularization terms, the L2 regularization and the L norm, which to avoid the embedding have exploding norm. Together, all those 4 parts is our loss for the embedding process. And the second part is a cross-network alignment process, which once we have 2 autoencoders and each generate embedding for 2 species, we use an idea inspired by natural language processing area, more specifically the language translation, where we can consider proteins as words and the also logs as words with similar meanings. So the autologs should have similar embeddings in the joint embedding space. So what we did is, for example, we have human and mouse. We have human and mouse autologs. We will first, for human autolog, we will first using the human encoder to generate an embedding. Since the human autolog's embedding should be similar with the mouse autolog embedding, we will then use mouse decoder taking the human autolog embedding as input and try to reconstruct the mouse topological information. And it also works the other way around. Combining those two directions will be our cross-training process, which is also the loss function for our alignment step. After iterating the individual network alignment process and the cross-species individual network embedding process and the cross-species alignment process several times, we will create a joint embedding that contain both within and cross-species functional similarity. And we will use cosine similarity, calculate the cosine similarity for each pair between two species. This is our prediction for how functional similarity two genes are in different species. Next, I will introduce our evaluation. The first evaluation is to see whether our predicted functional similarity is actually consistent with some gold standards. And the gold standard we are using is the gene ontology, which unifies the gene attributes across different species. And since the gene and the gene ontology terms has a many-to-many mapping relationship, in the right example here, we can see one gene can annotate it to more than one Go terms. So we use a G-card index, which is intersection divided by union, to determine how similar two genes are. So a higher G-card index between a gene pair implies higher functional similarity. So we rank the predictions created by our model Etna, and we compare the top pairs from our model to see whether the top pairs in our model also have a high functional similarity. And here is a score distribution for the top 5,000 predictions for our model versus 3 existing methods and 2 baselines. And this is aligning human to east. We can see our model have a higher G-card index, which means our model's predictions have a higher functional similarity than other methods and baselines. This is also consistent with other human with other model organisms. For human, fruit fly, and worm, our model is consistently better. And in the second evaluation, we are trying to use the joint embedding created by Etna as an input for other machine learning algorithms to predict some more complicated gene relationships. And here we use genetic interaction, more specifically the synthetic lethality, which is defined by when we perturb two genes, it will cause a lethal effect that we perturb either or one of the gene won't show that lethal effect. And we take, for a gene pair, we take the embedding for each of the genes, then we add the embedding together to generate input. And then we feed this input to a supported vector machine. And the supported vector machine will give whether the gene pair is synthetic lethal or non-synthetic lethal. And here we are using the information from 2 kinds of yeast, the Cerevisiae and Pombi, using a holdout part of the synthetic lethal information like as test set. We are testing whether using our embedding or one existing method, Monk's embedding, can predict the synthetic lethal information. The first task we perform is we use the information from two kinds of yeast and predict the holdout in one kind of yeast. And we can say our method is better than the existing method Monk. And the second task is that we are trying to use the information from one kind of yeast and predict the holdout in another kind of yeast, which actually achieves the translational study that we are aim at. And we can see our method is still consistently better than the existing method Monk. And it's also give a reasonable good prediction. And this shows using only protein-protein interaction information and using the embedding only created using the protein-protein interaction information and also log information, it can be used to predict more complicated genetic relationships. The final evaluation we did is to see whether our embedding can show some relationship between humans and human diseases and model organism functional pathways. And in this example, we rank top 1% of our ETNA prediction between human and mouse, and we clustered the data and identify functional modules. Then we do enrichment analysis with gene ontologies, drug targets, and human diseases to see whether those clusters are enriched with interesting disease or drug targets. And you can see there are lots of interesting clusters, but due to the sake of time, I'll only highlight one, which is bottom blue cluster that shows a cluster, the mental health gene with the drug target for the benzodiazepine, which is a drug that's designed for bipolar diseases. So for summary, our model is using an autoencoder as an algorithm and generate individual network embedding. And then we use a method inspired by natural language processing, then aligning the individual embeddings to the joint embedding space. And the joint embeddings are showed to have capture the functional relationship across species, and this can also be used for other machine learning algorithms to predict more complicated gene relationships. And finally, it can also identify connections between mouse functions with human diseases. For future steps, we are trying to analyze the dissimilarity in our ETNA score, because in the model organism study, especially the drug target studies, the dissimilarity is also very useful. The second, we are trying to use the imputed PPI network to expand the input size of our model. And third, we want to expand our model to work on weighted networks, especially co-expression network, so that we can do some more cell type or tissue specific analysis. And finally, because our alignment is bidirectional, this gives us the chance to do a multi-network alignment beyond just aligning a pair of species. So finally, I would like to thanks to my advisor, Dr. Vicky Yao, and also other contributors to this project, and also all the members in our lab, which gave me lots of project ideas and good feedbacks. And here is also a link to our preprint if you are interested. Thank you again.